This is Module 3, Transcription. In this video, I'll be discussing the aspects of transcription that are unique to eukaryotes, or cells that contain a nucleus. Eukaryotic transcription is somewhat similar to bacterial transcription in that it has similar stages. It's got initiation, elongation, and termination, and that there are a lot of proteins that work together to facilitate each of those three stages. But essentially, all the rest is different. So still adding to a 3' OH group for growing RNA strand, still complementary and antiparallel, but the process of starting transcription and elongating an RNA molecule is different between prokaryotes and eukaryotes. First of all, eukaryotes have nucleosomes. DNA is packaged and wrapped around nucleosome cores. And so before transcription can even begin, there needs to be some remodeling of the chromatin that is going to be expressed. So histone proteins need to be moved around, redistributed, so that RNA polymerase can actually bind to the promoter. Also, the promoters in eukaryotes are completely different. It's not just a single RNA polymerase enzyme doing some work with a sigma factor. It's a whole suite of enzymes, proteins, subunits, all working together to do this work. Also in eukaryotic transcription, transcriptional activator proteins are responsible for initiating this process or starting this process. And there's a good variety of transcriptional activator proteins also. There are 3 different types of RNA polymerase in eukaryotes. RNA polymerase 2 is akin to the one in bacteria in that it does mRNA synthesis, so it makes your messenger RNA that is eventually made into proteins. But the other 2 promoters do a little bit of different things. So polymerase 1 and polymerase 2 is what they're called. Polymerase 1 is responsible for synthesizing ribosomal RNA, and polymerase 3 is responsible for synthesizing transfer RNA. So for each of the 3 major RNA players, messenger RNA, transfer RNA, and ribosomal RNA, there are 3 different polymerases responsible for creating each of those molecules. Thinking along the lines of evolution again, when the transcriptional process in archaea was examined, there were some notable similarities between how archaea transcribe their DNA into RNA and how eukaryotes do this also, suggesting that there's actually a closer ancestral relationship between archaea and eukaryotes, whereas previously it was thought that archaea was more related to bacteria. Their RNA polymerases are similar in structure and function between archaea and eukaryotes. Archaea promoters have a lot of the same consensus sequences found in eukaryotic promoters. Archaea regulatory regions have a lot of the same sequence similarities as well. And so that means bacterial transcription is unique compared to eukaryote and archaeal transcription. The promoters of genes transcribed by RNA polymerase 2, which is transcribing mRNA, consist of a core promoter, where there's lots of consensus sequences, common to most protein coding genes, and then a regulatory promoter that contains consensus sequences that are fairly unique to a particular gene, or the combination of sequences in the regulatory promoter are unique. And so the core promoter helps RNA polymerase to bind to its DNA template and then elongate to make RNA. But the regulatory promoter controls whether or not the polymerase is able to bind in the first place. We'll come back to that also. So let's look at initiation in eukaryotic transcription. This is accomplished by RNA polymerase 2 and a whole host of transcription factors. One of those transcription factors is called a TATA binding protein, and this is the protein that binds to the TATA, the TATA box, in the core promoter region. Okay, so I promised you it was complicated in eukaryotes, and here it is. Transcription is initiated at RNA polymerase 2 promoters, and so RNA polymerase 1 and 3 have slightly different consensus sequences in their promoters. This is only initiated at these promoters when specific transcription factors called TFID bind to the title box, followed by the binding of a pre-assembled holoenzyme, which would be this image right here, that contains all of your general transcription factors, the TF ones, and it also contains and then it's protein base. It's a protein enzyme. So all of these transcriptional factors have to combine with this RNA polymerase II to form a mature holoenzyme. Once the holoenzyme is finally bound to the core promoter, now it's bound to the core promoter, specifically at the TATA box, which is facilitated by a TATA binding protein. Something kind of bizarre and strange happens here. So right here we have our RNA polymerase holoenzyme. So lots more than just RNA polymerase is stuck into here. And then we have this molecule, which is called a mediator. This mediator is capable of binding to proteins at the enhancer site, which is actually like all the way out here. And so what happens is when the core promoter is present, when the holoenzyme is present and it has its mediator attached, the DNA upstream will bend to associate this transcriptional activator protein with this mediator protein. And so without the mediator, this interaction wouldn't work. And then enhancer proteins do exactly what they promise. They stabilize the RNA polymerase on the DNA template and ensure that it will go all the way to the end of the template. Now let's look at the elongation stage. Eukaryotic RNA polymerase II functions pretty uniquely during elongation. DNA in the double-stranded helix enters in through a channel, like a small groove, and then unwinds in the core of the protein. When it unwinds, this leaves one strand of it open to transcription by the remaining subunits of the enzyme, which are catalytic. And so the DNA double helix enters through this groove, unwinds here, and then the DNA RNA duplex is, so this basically means when RNA is being made, you have a DNA RNA duplex. When this happens, it's bent at a right angle, which positions the 3' end of the RNA at the active site of the enzyme. So the way that the DNA enters this enzyme, basically the enzyme turns it and positions it so that the OH on the 3' end is in the perfect position for adding a nucleotide. And then new nucleotides are added to the 3' end of the RNA. Termination in eukaryotes, fairly similar. RNA polymerase I uses something similar to the Rho factor in bacteria. RNA polymerase II uses hairpins, which is similar to the Rho-independent method in bacteria. And then there's RNA polymerase II, which has a unique type of termination. So RNA polymerase II is right here transcribing its DNA target. And these enzymes tend to just keep keep transcribing until they get, they fall off or they get to the end or something like that. And so we have to tell it when to stop. So it's not making all of these nucleotide, all these RNA molecules for no reason. So as the mRNA is exiting the RNA polymerase II, there is a cutting event that does double-stranded break near the end of the RNA molecule. And this is considered the pre-mRNA, and this is what will be processed and used to create a protein. This does not stop RNA polymerase from continuing to transcribe. When this piece breaks off, a protein called RAT1 endonuclease, which is an enzyme capable of degrading nucleotides, RNA, attaches to the 5' end of this RNA that's sort of right now useless because it shouldn't have been transcribed in the first place. When this pre-mRNA moves towards the RNA polymerase, it degrades the RNA as it goes. And then when RAT1 reaches the polymerase, transcription is terminated. And so this too is sort of like a little Pac-Man. He binds to the 5' end of the growing RNA strand and then basically chops it up and degrades everything as it goes. And then when it bumps into the polymerase, it falls off.